Okay, guys, welcome to Chapter 24. We're going to talk about the lymphatic system and immunity today. This is essentially two of the systems that keep us disease-free. They work hand in hand. When we're talking about the lymphatic system, we're talking about a specific set of structures and vessels that we're going to refer to, and we'll talk about where those organs are located and what they do. And when we talk about immunity, we're talking really about a broader set of cell types that are stationed really all over the body that essentially patrol all our tissues, all our fluids, and scan them for pathogens. And if they encounter them, they will seek out and destroy them so that we do not come down with all manner of diseases. So when we talk about immunity, we can talk about different types of immunity, different flavors of immunity. Innate immunity is fast, nonspecific, and it has no barriers, or no memory, rather. And what we mean by this is that we use the same manner of chemical attack to knock the pathogen out, regardless of the identity of the pathogen. Examples of nonspecific immunity include our external body barriers, such as our skin and our mucous membranes. The pH extremes that we have on these surfaces, which are hostile to the growth of bacteria and mold and so forth. Phagocytes and natural killer cells, which essentially wander around the body's tissues, gobbling up anything that it does not recognize as belonging to us. And the way that the immune system does that is by checking a very special type of protein called a major histocompatibility protein. It's like our chemical ID tag, and if the invading cell doesn't feature this very important identifier, the natural killers in the phagocytes will gobble them up and then will fuse those contents with the lysosome and destroy them. Fever basically is a way of cooking out the pathogen and also stacking the deck in favor of conditions that are designed to enhance the replication rate of our white blood cells. And then the entire inflammatory response, which is we're going to find out features the area around the wound or the infection heating up, swelling up, hurting, and turning red. And this is all designed to bring blood flow to the area, and in that blood are the white blood cells that are going to help us battle the infection, as well as cells that are designed to patch the wound after the infection has been knocked away. Complement is a type of nonspecific chemical attack that blows holes in the cell membranes of invading pathogens, and interferon is a type of antiviral chemical attack. All these chemical attacks, and inflammation you could kind of categorize as a chemical attack, are not specific to the type of pathogen. They follow the same pathway regardless of whether or not the pathogen is a bacteria, a mold, or a virus. Adaptive immunity is the kind of immunity you probably think of when you think of having kicked a particular infection. It's slower, specific, it has a memory. It's mediated by lymphocytes. These are T and B cells, and what they are, they're a type of white blood cell that has the ability to recognize a specific pathogen and take him out using an attack designed only for that particular strain of bacteria, mold, fungus, so forth. So let's talk about the lymphatic system. Lymphatic tissue is basically reticular connective tissue that contains lymphocytes. Remember, lymphocytes are a type of white blood cell. Bone marrow is the source of these lymphatic stem cells. Basically, hematopoiesis has as one of its jobs the production of all the formed elements of blood, and among those are the leukocytes, the white blood cells. Lymph is the interstitial fluid in the lymphatic vessels. It returns excess filtrate from the capillaries to the circulation. It also helps to transport dietary lipids directly through the walls of the small intestine right into special structures called lacteals. This is a non-immune function of the lymphatic system. And it's also involved with maintenance and distribution of these lymphocytes via the lymphoid organs. It also helps to filter bacteria and activate defenses. Now, if we look here at the lymphatic system, you can see that it's made up of specific organs. You have here the bone marrow, which is engaged in hematopoiesis, and as a result generates the white blood cell. These green lines here, these are lymphatic capillaries. This is the spleen, okay? In the small intestine, you have clusters of lymphatic tissue called Peyer's patches that lie just below the lumen of the small and large intestine. And what they're designed to do is to catch any pathogen that crosses the walls. You've got the thymus behind the sternum and in front of the heart, and it's the site of T-cell education. It's very important in our childhood, but it involutes and becomes a mass of scar tissue as we age, as it becomes no longer necessary. And then these little sort of green pea-looking things, those are lymph nodes, and lymph nodes are sites of filtration of body fluid where we scan them for the presence or absence of pathogens. If we detect them, we then engulf them, chop them into little pieces, and as a result destroy them. But more than that, we present those little pieces on the surfaces of the macrophages, the antigen presenting cells, or APCs, and this alerts the lymphatic system to the fact that we have a pathogen that has now managed to make it deep into our body tissues, and we should, as a result, recruit a team designed specifically to take that pathogen out. Okay, lymphatic vessels begin at lymphatic capillaries. They're slightly larger than blood capillaries, and they have overlapping cells that act sort of like a one-way valve, so that when the pressure is higher outside the lymphatic capillary than it is within the lymphatic capillary, this will force fluid into the lymphatic capillary and eventually, through a series of valves, make it all the way to a lymph node where the tissue fluid will now be examined for the presence or absence of pathogens. They eventually merge to form larger vessels, which are thin-walled and have more valves than veins do, and they periodically encounter the lymph nodes, which contain lymphocytes in an encapsulated structure. And what you'll notice about a lymph node when we get a chance to look at one is that it has more incoming vessels than it has outgoing vessels, and that's designed to slow the flow of the tissue fluid through that organ to give the lymph node time to filter and take out any pathogens that might be present, and then process them and present them for the rest of the immune system. As they exit the lymph nodes, they enter larger and larger lymph vessels called ducts. These eventually lead to the thoracic duct, which leads to the left subclavian vein, which basically joins up with the jugular vein, and the right lymphatic duct, which eventually joins up with the right subclavian vein. And at that point, this cleansed tissue fluid will now rejoin the rest of the circulatory system and eventually become part of the blood plasma. So what we find here, when we look at this arrangement of vessels, you can see here that generally where you find a nerve, you will find a lymphatic vessel. You'll also find an artery and a vein. They tend to run together, and that's for the structural integrity of the body. But what you find is that this is one of the hidden benefits of exercise, where the more rapidly we move blood around the body, the more rapidly we're going to push fluid through the lymphatic system, the more effectively it's going to be cleansed of pathogens. And so this is one of the benefits of both lymphatic massage, which you are schooled in, as well as just regular old physical activity. It keeps us disease-free because it keeps these fluids moving around the body. And what you're looking at here is just the relationship between the flow of blood through the arteries and the veins and the capillary beds and the flow of lymphatic fluid through the capillaries. basically, when the interstitial pressure is higher than the pressure in the lymphatic capillary, and the lymphatic capillary isn't blocked, the fluid will flow into this blind sac, and now it'll be inside a lymphatic capillary, which will eventually lead to a lymph node. Okay, so you can see how these one-way valves only open inward, allowing this interstitial fluid and its components to move in. So this is not a very sophisticated filtering mechanism. We sort of want everybody to come in here if they can, regardless of their size, because we eventually want these cellular components and these particulates to encounter a lymph node and then be addressed accordingly. Okay, so looking at the basic setup for how lymph flows around the body, what you can see here is that at the tissue fluid, what happens is that lymphatic capillaries gather up this uncleansed interstitial fluid, and it now enters all these lymphatic capillaries. Eventually, we make it to one or several lymph nodes. What you'll find, if you look back in your book at the diagram showing you the lymphatic system, is that these lymph nodes are stationed in areas of the body where they're most likely to encounter an infection. We have a high concentration of lymph nodes in the inguinal region, in the axillary region, the popliteal region, and lining the region around the lower GI. And these are all areas where we would expect a pathogen possibly to enter. As we move through these lymph nodes, the fluid gets cleansed, and now we enter what we call lymphatic vessels. These are larger in diameter, and again, the flow is governed by one-way valves, and eventually we make it to either the right or the left subclavian vein, at which point we jack back into the venous circulation, and we are ready now via our passage through the lymphatic duct to enter the circulation again at large, and then the heart will pump now this fluid around the body. Okay? So let's talk a little bit about these lymphatic organs. The primary lymphatic organs contain stem cells. Remember that a stem cell is a cell that has a multitude of fates open to it. They divide and develop into mature B and T cells. They do this through the process of differentiation. The B and the T cells are very important. They're generated in the red bone marrow and the thymus, and what they do is they modulate our humoral and our cell-mediated immunity. Now you might wonder, well, what is humoral or cell-mediated immunity? And basically, humoral immunity is that type of immunity that is mediated by antibodies. Antibodies are basically magic bullets designed to seek out and destroy a particular pathogen by binding to it, whereas T cells are tasked with actually showing up at the tissue that's diseased and destroying it. Okay, we'll take let's talk about the secondary organs. This is where the immune response occurs. This happens in the lymph nodes, the spleen, and the lymph nodules. And what happens here primarily is that we alert the immune system to the presence of these pathogens, and now we're able to basically generate a team of white cells designed specifically to take out that particular pathogen by producing antibodies either that bind to them very effectively or T cells that are designed to interact with only their antigens and as a result destroy them. Okay, so the thymus is one of the most important lymphatic organs early on in the development of the lymphatic system. It has two lobes, and it's located behind the sternum and anterior to the heart. It contains T cells which divide and basically learn how to become competent in the immune system while they stay inside this organ. You could think of the thymus as T cell school, and in fact the term T cell comes from the fact that it matures in the thymus. Essentially what happens in the thymus is that we select cells that are capable of responding to pathogenic attacks while tolerating our own tissue. So this is very important because if we don't weed out those cells that are tolerant of our own tissues, what will happen is that we will generate an autoimmune response. We'll actually end up attacking and destroying our own body tissues and cells because we haven't properly screened this population of leukocytes. So self-reactive cells are removed, and competent cells are produced, and cells that weren't competent before but are tolerant are now activated. So this is a series of chemical interactions that occur inside this organ that now render this cell population capable of going out now and populating the other immune organs. Now, what are these other immune organs? Well, they include the lymph nodes. You find these scattered all over the body. They're concentrated in the mammary glands, the axilla, and the groin, as well as around the popliteal region and clustered near the GI. They contain mature B cells and T cells, as well as dendritic cells and macrophages. Now, these cells are important because these dendritic and macrophages are what we call antigen-presenting cells, and they have the capability of engulfing a pathogen, chopping them up into little pieces, and then presenting the component parts on the cell membrane in combination with an MHC protein. And this is what serves to alert the rest of the immune system, the B cells and the T cells, to the presence of a particular pathogen inside the body. Other processes that go on inside the lymph nodes include filtering, trapping, processing foreign substances. Macrophages and lymphocytes destroy most foreign substances, but also, more importantly, they present them to the rest of the immune system. And so here you can see a lymph node. And this is what I was telling you about when we were discussing the flow of fluid through this organ. You can see here that there are more afferent vessels than there are efferent vessels. And what this ensures is that the fluid will slow down as it enters this organ, and there will be time taken to adequately process now any pathogen that might be present in this fluid, and engulf and destroy any pathogen it might encounter. In addition to that, as we stated before, we alert the rest of the immune system to the presence of essentially strangers in town. Now notice that one of the other things that we feature in this diagram are the valves, and these valves also ensure that the lymph only flows in one direction, which is into the lymph node through the afferents and out through the lymph node through the efferents. And what types of cells do you find in here? Well, dendritic cells and macrophages, which are essentially antigen-presenting cells. And then these other cells, the B cells and the T cells, are part of our specific immunity. And plasma cells are essentially what a B cell becomes once it's been activated and is actively in the process of secreting an antibody, which again we can think of as a magic bullet designed to take out one particular pathogen. Okay, the spleen is located between the stomach and the diaphragm, and it contains blood-filled venous sinuses and red blood cells, as well as macrophages, lymphocytes, plasma cells, and granular leukocytes. So this is an interesting collection of tissue. One of the spleen's job is to recycle worn-out red blood cells, as we discussed when we were talking about the circulatory system. But it also has an immune function, and in the immune function, what we have again are antigen-presenting cells, the macrophages, but we also have the lymphocytes and the plasma cells. Remember, those are part of our specific immunity. And then we also have granular leukocytes. Now, what the heck are granular leukocytes? Well, a granular leukocyte, or a granulocyte, is simply a white blood cell that contains little tiny membrane-enclosed bubbles that have chemicals that are designed to destroy a pathogen if it shows up. And so you could think of this as your body's version of pepper spray, okay, or chemical mace, designed to take out a particular pathogen. The granulocytes are part of our non-specific immunity. The granulocytes are the eosinophils, the basophils, and the neutrophils, and they all contain chemical components designed to neutralize an invader. The act of them spraying the invader with these chemicals is called degranulation. Okay, so that's what happens when they dump their load. The spleen also destroys worn or defective blood cells and platelets. Remember that platelets are involved in the blood clotting process. It also stores platelets. It attacks foreign substances in the blood, and it's also the site of fetal hematopoiesis, meaning the manufacture of blood during fetal development. Okay, let's talk a little bit about our nonspecific barriers. This is our innate immunity. This is our protection that is the same regardless of the type of attack that's being used to make the body sick. We don't care about the identity of the pathogen here. We use the same approach regardless of his category. Okay, so the skin is one of these examples. It is both a physical and a chemical barrier. The epidermal structure and the constant shedding keep organisms from settling down and growing long term on the skin. So we constantly are changing now the surface of the body by shedding these cells and not allowing any pathogen to set up for very long. The mucous membrane have a sticky mucus layer that traps microbes and other pathogens, and the cilia that move it up and out. Basically, remember that the mucociliary escalator, which is located in the upper respiratory tract, constantly beats mucus that traps these pathogens towards the oral cavity, where they can either be spit out or swallowed. And once they're in the GI, the digestive enzymes and also the immune cells that line the small and large intestine, the Peyer's patches, will take care of it. There's also fluids that we secrete: tears, saliva, perspiration, and nasal secretions, which dilute the bacteria, and they also have antibacterial activity in them. There's an enzyme called lysozyme in saliva and tears and sweat, which is designed to weaken the cell walls of pathogenic bacteria, and as a result, when they encounter an osmotic shock, they will either shrivel up or burst and become inactivated. We also have the movement of fluid through certain compartments in the body, such as the flow of urine, the flow of solid waste, which is defecation, and vomiting, and also sweating. And this is a way of clearing out the system. Let's talk about our internal defenses. Interferons are antiviral proteins. They interfere with viral reproduction inside the cell by preparing neighboring cells for a viral attack, causing them to secrete or produce enzymes, rather, that will destroy viral intermediates in the cytoplasm. Now, interesting thing about interferons, they were thought at one time to be a miracle cure for viruses, and they were manufactured with the intent of destroying viral disease in the late 80s. The problem, as it turns out, is that megadoses of interferon is very hard on the liver, and as a result, these experimental drugs were pulled off the market because the cure was worse than the disease in many cases. So there's probably a reason why, when the body makes these components, it makes them in very small amounts. The complement system enhances immune attack. It causes cell membranes of pathogens and diseased tissues to rupture. It also attracts phagocytes, and it tags microbial cells for destruction. So this is a way of improving the action of specific immunity. So complement relies on specific immunity in order to operate, but again, the mode of attack, the chemical strategy is the same regardless of the identity of the pathogen. Transferins bind iron and they starve bacteria, and there are also antimicrobial peptides that can lyse microbes. And examples of these would be things like perforin, okay, which pokes holes in microbial membranes and causes them to bleed out. Phagocytes are specialized to ingest microbes and cellular debris. Examples of these are neutrophils and monocytes, which differentiate into macrophages. And this type of defense has a two-pronged effect. It both destroys the pathogen by ingesting him and breaking him into little pieces, but it also warns the immune system of the presence of a particular pathogen by displaying these digested bits on the surface of the cell membrane. Okay, so this is, again, a two-pronged strategy to rid us now of disease and illness. 5 to 10% of these lymphocytes are what we call natural killer cells. They destroy microbes and tumor cells, and they're present in the lymph nodes and the red bone marrow. And essentially what they do is they show up at the site of infected tissue, punch holes in it, and cause it to die. The natural killer cells are part of a very important function of the immune system called immune surveillance. Basically, the immune system, the cells of our nonspecific and specific immunity, sort of walk the beat of the entire body looking for anything unusual. And if they find it, if they find a strange antigen presented on the surface of a particular cell or tissue, they'll move in and destroy it. Now, this includes not only tissue that's infected, but also tissue that can become cancerous. And the strategy here is that by recognizing these aberrant cell types, we can knock holes in them, destroy them, and cause them to be inert. Okay, inflammation is the body's response to injury and infection. The damage leads to mast cells, basophils, and platelets all releasing histamine, which has as its effect to increase the permeability and vasodilation present in blood vessels. This is why when we have an inflamed area, it swells up and it gets red, okay? Because what's happening now is we're bringing more fluid to the area. A lot of that fluid is blood, and the redness indicates that there's blood coming to the area, and the swelling, the edema, indicates that there's fluid coming to the area. Now, one of the other outcomes of this is an increase in temperature, which causes now the environment in which the pathogen is going to attempt to reproduce to become hostile to that process. So basically we try and cook the pathogen out before he gets a chance to cause any disease. But this elevated temperature isn't bad for the white blood cells that are designed to fight the infection. So we're sort of stacking the deck in favor of our emergency med techs, the white blood cells. Another thing that happens with inflammation is that it hurts, okay? And that is the body's way of telling you that you should not use the affected organ, okay? The idea behind this is that you have to keep from actively using the affected area in order to give these white cells a chance to affect both the conquering of the pathogen and also to begin effective repair of the pathogen. So again, a two-pronged method in order to deal with now the incumbent infection. You also have leakage of clotting proteins into the tissue. This tends to isolate the bacteria behind a clot. So we sort of box him in. Now we've got him cornered and we can take him out. Inside this sort of blocked off area are white blood cells, and they have the ability to attack and destroy the pathogen. Phagocytes are attracted to the site. The neutrophils and macrophages eat either the dying tissue and or the pathogen and cause them to be destroyed. And then we create here a pocket of dead cells that we call pus, which moves to the body surface or into the cavity, and as a result is cleared. So anytime you see pus, that's a good thing, because that's your body actually fighting the disease process and effectively clearing out either dead or dying tissue or the pathogen itself. And so this is just showing you the inflammatory process. You have here tissue injury resulting now in the release of chemical signals that attract white blood cells directly out of the circulation. They're drawn to the area. They then engulf any disease-causing organisms. And then after that's done, what happens is that we begin the process of repair. Fibroblasts come in and begin to lay down scar tissue, and eventually the block that was originally covered by a clot is now covered by scar tissue. And so we've stanched the flow of blood, and we've essentially performed a biological patch job on the area. Once the damaged tissue has been cleared away, the pathogens have been removed, the white cells then jump back into the circulation, and they head off to their next destination. Fever is an abnormally high body temperature. It results from chemicals being released called pyrogens that cause the hypothalamus to set now a higher body temperature. So this is the new set point of the thermoregulatory system. The normal temperature control action has now a new set point. It is stimulated by toxins or internal signals, these pyrogens, one of them being interleukin-1, which is one of the chemical signals that are released by white blood cells when they're activating each other. And it also has the effect, again, of raising the body temperature. And what does this do? Well, it causes conditions to be created that enhance the reproduction of white blood cells, and it also basically cooks out the pathogen, causes it to be too hot for him to reproduce, and as a result, you don't have as many pathogens to battle. Okay, so fever, a very important strategy that the body uses to try and, again, rid you of infection. Okay. Adaptive immunity is specifically directed against a particular type of invader. Now, this is what we mean when we're talking about the lymphocytes. The lymphocytes they all do a similar job, but they don't do it in exactly the same fashion. Okay? This involves a cell or an antibody directed against a particular antigen, which can be any substance or a microbe or a food pollen or a tissue. Okay, anything weird. Okay, it can be living or non-living. Doesn't make any difference. As long as it has a molecular display that's not like our body tissue, you're going to warn the lymphatic system that there are disease-producing entities in the vicinity. Normally, these lymphocytes are tolerant of our own tissue, meaning that they will not attack and destroy normal body tissue. So when the immune system is working as it should, what happens is that when foreign substances enter the body, they're identified as not belonging to the body proper, and we recruit T and B cells that are designed to specifically interact and bind very tightly to antigens that are present on the invader. And what this results in is a selection of those particular white cells, those particular lymphocytes, that interact effectively with the invader, and their population is amplified. Basically, they're told to replicate, to duplicate themselves. And so what we generate here is a task force that has profiled the invader, and now they're going to go out and attempt to destroy it. How do these T and B cells come to be? How are they generated? Well, they come from stem cells in the red bone marrow. B cells mature in bone marrow, while T cells are generated in bone marrow but migrate to the thymus, where they are now selected for two very important properties: tolerance and competency, meaning they're capable of moving throughout the body without attacking our own tissue, but they're capable of attacking a pathogen if he shows up. Okay, so two very important characteristics that each lymphocyte has to meet while it's in the thymus. During maturation, both make particular proteins in plasma membranes. These are called antigen receptors, and this is what actually interacts with whatever the strange molecule is that's on the surface of the pathogen. And so when you talk about the lymphocytes, one of the things that you have to understand is that they're all a little bit genetically different, and that genetic difference comes in the form of the genes responsible for creating the antibodies and the genes responsible for creating the T cell receptors. Basically, there's millions and millions of different flavors of these antibodies and receptors in an operating normal immune system. And as a result, we have the ability to pick out from all those different white cells the particular white cell that is going to recognize, bind, and destroy our particular pathogen, our unwelcome visitor. Okay, in cell-mediated immunity, the T cells directly attack the, the, the infected tissue. Okay, they show up at the site of the infection and destroy the tissue directly. This is done by killer T cells. In antibody-mediated responses, the B cells have now been alerted to the presence of an unusual antigen in the body, and they differentiate into plasma cells, which are basically antibody factories. One of the things that you'll notice when you look at a plasma cell under the microscope is that it has an extensive endoplasmic reticulum, and that's because it is making a lot of protein. Okay, remember that the ER is basically the protein producing factory inside living tissue, inside living cells, and so the more protein processing and producing you do, the more ER you're going to need. And so these plasma cells have an extensive rough ER. And so we're cranking out these antibodies that then move through the body's fluids, through the plasma and through the tissue fluid, and eventually they were going to encounter the pathogen in question, bind to it, tag it for destruction, and take it out. Helper T cells aid both the cell and antibody-mediated responses by producing chemicals that make the B and T cells designed to attack that pathogen multiply more aggressively. Okay, the major histocompatibility complex is that cellular ID tag we were talking about. These are self-antigens on the surface of all the cells of our body. They're unique to each individual with the exception of identical twins, and they allow T cells to recognize foreign material. The antigen triggers plasma cells to produce antibodies, which are basically Y-shaped magic bullets that are designed to bind very tightly to the antigen in question. The other end triggers recognition by phagocytes, and this helps us to destroy the tagged antigen. So this is the magic bullet. This is what it looks like. And the region that binds to the bad guy is this region in the box. Okay, these are called the variable regions. And it affects the destruction of the pathogen in several different methods. You can have the antibody bind to the surface of the pathogen, and that will attract components of the complement system, which will enhance the phagocytosis of the entire complex. You can also, by activating the complement system, produce a cascade of chemical reactions that ultimately results in the lysing of the pathogen by punching holes into the cell membrane. The other way that the antibody can work is that it can bind pathogens to other pathogens. And essentially what happens then is the molecular weight gets so high after a while that the entire complex falls out of solution. And this is a process called immunoprecipitation. And essentially now what happens is that the pathogen is sort of stuck in the mud, and now he's an easy victim for a wandering macrophage to come by and pick him off, because now he can't invade any of the other body's tissues. And it's just showing you sort of how this works, right? This is the antibodies binding to the antigens, and here you can see, right, two pathogens, these little kidney-shaped bad guys here, are bound to each other, and you can imagine that you can bind 2 to 3, 3 to 6, 6 to 12, and eventually what happens is that the whole complex gets so large that the whole thing just falls out of solution and is easy pickings for a wandering macrophage. Okay, so how do we trigger this adaptive response? Well, we have to recognize the foreign antigen to begin with. B cells can find it anywhere in the body. T cells have to have it presented in combination with a major histocompatibility protein. So it has to understand it in that context. You could think of the MHC as almost like an hors d'oeuvre tray and the antigen as being the hors d'oeuvre. Okay, and if we sample the hors d'oeuvre tray and we recognize it, that combination of tray with hors d'oeuvre, then we become activated. Antigen presenting cells are what do this. Antigen presenting cells are all over the body. You've got them under your skin, you've got them in your Peyer's patches, you've got them in all your lymphatic organs, okay? And they're wandering around, okay, looking for bad guys to engulf and show to anything that is a component of the lymphatic system, right? You can have wandering macrophages and you can have fixed macrophages, and they both do essentially the same job. They gobble up the bad guys, chop them into bits, and then show them to the lymphatic system for the lymphatic system to ultimately generate now its attack. Antigen-presenting cells include macrophages, dendritic cells. These interact with B cells. B cells also have the ability to ingest and display antigens. You've also find them in the respiratory, the GI, the urinary, and reproductive tracts, as well as, obviously, in the lymph nodes and lymphatic organs. Antigen-presenting cells ingest and digest the bad guy into little pieces in vesicles. They synthesize MHC protein and pack it into the vesicles. The two vesicles then fuse, and the antigen fragments bound to the MHC are inserted into the plasma membrane, and this is the warning signal that tells now the immune system that there is a stranger in town. So here we can see this process in action. So here is our antigen-presenting cell. Here he is either stapled to a particular organ or tissue, or he's wandering around, okay? And so what does he do first? He engulfs the bad guy, okay? Next thing he does is he digests the bad guy into little pieces. Next thing that happens is that we synthesize MHC2 molecules. We then fuse now the MHC molecules with the digested peptide fragments, and then this whole complex is now going to be moved towards the cell membrane, where the MHC complex and the antigen are presented together. Okay, here's the key up here. These are the fragments. This is the antigen peptide. Okay, and then this is the MHC2 protein. And it's this combination of antigen and MHC2 that the lymphocytes understand and use as a way of generating their attack on the particular invading organism. Okay, T cells also need a co-stimulator, and this is interleukin-2, binding both to a helper T cell and receiving the interleukin-2 chemical stimulation produces a response that makes the T cell begin to rapidly divide. This forms a large population of carbon copies of that particular T cell, and now it goes off and either directly destroys the tissue, or we can form as a process that's ongoing during this response memory T cells that basically stay with you for the rest of your life. The same is true of the B cells. The helper T is essentially a facilitator, and what he does is he enhances the recruiting and the amplifying of the particular B and T cell population that's designed to pick off the invader. And those cells will actively fight the invader, but they'll also form memory cells during this process that sort of sit around waiting for the next time that particular pathogen shows up again, and if it does, they're there to destroy it. Okay, cytotoxic T cells destroy cells that are infected directly. They work against tumor cells and infected cells, as well as transplanted cells. They also form memory T cells during this process that hang around for sometimes your entire life and give you a rapid response to infection. And so what you're looking at here is just this process. Okay, here we have an infected body cell. It's one of the properties of our body cells that we have the ability to engulf, process, and present these little bits of pathogen on the surface as, again, a warning signal to the immune system that will then interact with it. Okay, here we have a cytotoxic T cell interacting with a presented antigen. It also will interact with a helper T cell, and the result now will be an activated cytotoxic T cell that will begin to divide at a high rate and will go off now and attack any infected tissue. Okay, so we have to pick out our winner here. Okay, this is our winner, our T cell that's going to recognize this particular antigen, and then we have to tell them to go off and multiply and fight the infection effectively. Okay, B cells are found in lymph nodes, and they respond to antigens. They respond more rapidly if it's presented with an MHC protein, but they can also interact directly with an antigen on a pathogen. With the help of IL-2, they will enlarge, divide, and become a clone of plasma cells, which will release antibodies that will bind the antigen. Some of these will remain as memory B cells in our body for the rest of our lives. These are ready to respond quickly if the antigen is ever met again. And so this is basically the biological example of what doesn't kill you makes you stronger. Okay, if you're able to fight an infection, actively knock it back through the normal functioning of your immune system, what you store is large numbers of memory cells, and then the next time that pathogen shows up, we're able to take him out before he ever gets a chance to make us sick. So often people who are very sick as children are very healthy as adults because they've survived all of these attacks, and as a result, they're not prone to reinfection by the same pathogen. Now, if it's a different pathogen, then we have to go through this entire process again. Okay, here you can just see how this works. This is an activated cytotoxic T cell, and what it's doing is it's presenting now its receptor, its T cell receptor, that has interacted with now the presented antigen. Okay, so this is our infected body cell saying, help me, help me. Okay, the T cell comes by. He now sees this complex presented on the surface. He will then produce enzymes that are designed to destroy this particular infected cell. Okay, and now what happens is that phagocytes come by and clean up the mess. Okay. Over here we have the activated cytotoxic T cell interacting with an infected cell and producing now granulysin and perforin, which causes the cell basically to bleed out. Okay, and as a result, he's no longer viable. So in one case, we attract phagocytes to the area, and in one case, we cause the self-destruction of our target. Okay, chemical attacks by cytotoxic Ts that again have to show up where the infection is taking place in order to deal with it. And this is just showing you the difference between the primary and the secondary response to an infection. Notice that in the primary response, the IgM, which is the first class of antibody that's produced, will peak at around a week, and the titer is going to be relatively low. Okay, this will be followed by a peak in the titer of IgG, which is that Y-shaped antibody we were looking at earlier, and this guy is really designed to do the lion's share of the attack of humoral immunity on the pathogen. And then once the pathogen's knocked out, these levels will drop back down. Okay, there will be certain low circulating levels of IgG in your body throughout your lifetime. Okay, but essentially now that the infection is passed, we don't need to keep cranking this out. And you might think, well, how does that happen? Why do we quit making the antibody? Well, because there are also populations of lymphocytes that are made during this initial response, this primary response, they're like brakes. And what they do is they inhibit the activity of the T and the B cells after the infection is passed. Now, when the same pathogen shows up a second time, what happens here is that the response is so much more rapid and it's so much higher in magnitude that we don't get a chance really to produce disease. So we take the invading pathogen out in very short order, and we never even know that we were exposed. Okay, there are different types of antibody class actions. A neutralizing antigen will bind and neutralize a toxin. They can also immobilize bacteria. They can agglutinate pathogens by connecting them to each other and causing phagocytosis to happen more easily. And you can also activate complement and enhance phagocytosis, which will attract phagocytes to the area and gobble up the whole mess. Okay, memory is, again, what we were talking about earlier, the fact that during the course of this response, we generate cells that are designed to, again, fight with the pathogen if it shows itself again. These are long-lasting. They produce antibodies in the case of B memory cells, and in the case of T memory cells, they're just sitting around waiting the next time that the pathogen shows up. So you've got a lot of sensitive memory cells. They're much larger, and they have a quicker response time. This leads to the secondary response. The primary response can be naturally required, or it can be artificially acquired by vaccination, where we have killed cells, our isolated antigens from pathogens, which is really the majority of vaccines that we use today, or portions of viruses, which we also generate using recombinant DNA technology. We don't actually take live virus and isolate these particles much anymore. We synthesize them artificially. But essentially what we do here with vaccination is we fool the immune system into thinking that a pathogen has invaded you. And instead of actually giving you the pathogen, we just give you his ID tag, and now during the primary response, what we generate now are memory cells, and then the next time that pathogen shows up, we can take him out in short order. Now, as we age, the thymus will atrophy, which isn't a big deal because we don't really need it once we have generated a competent T cell population. We have fewer responsive T cells, however, and as a result, we have a poor B cell response, and as a result, a poor response to new infections. So this is one of the dangers with aging, is that we're not as capable of fighting off a pathogen if it shows up, and as a result, we're more likely to get an infection and to get sick. So this is why in the field of gerontology, we have to take special care to treat wounds, injuries, and infections in a relatively rapid fashion, so that we don't have the possibility of septicemia or of the formation of responses such as ulcers, okay, which are generated when we basically cut off the blood flow to an area of skin and cause that skin to necrose, and as a result now breach that nonspecific barrier, and we can now introduce pathogen into the body tissues and into the blood. So this is one of the challenges, again, for people who are entering the medical profession, because one of the largest needs populations that's going to come through as a result of the baby boomer bubble are aging baby boomers. And so you're going to have a large number of people entering, the latter years, 65 and up, and an effective application of gerontological techniques is going to produce now a much more beneficial outcome as our population dynamics shift. So this is one of the responsibilities with which you are tasked, which is learning how to take care of the old folks so that we have a much better quality of life as we age. All right, I will see everybody in class. Thanks for listening.